Hi, my name is Venkatesh. Today, I'm going to tell you about enzymes involved in cloning process. Enzymes are used in cloning to collect gene of interest. These enzymes cut the gene of interest required for gene cloning. Types of enzymes: nucleases, polymerases, ligases, transferases, DNases, RNases, phosphatases, kinases, restriction endonucleases. Nucleases. These enzymes are used to cut phosphodiester bonds present between nucleotides. Nucleases are of two types. Exonucleases cuts phosphodiester bonds towards N. Exonucleases are of three types. Lambda exo 3 Bal31. Lambda exo. These are the nucleases which cuts phosphodiester bonds towards 5' and these nucleases are isolated from lambda of bacteriophage. XO3. These nucleases cuts phosphodiester bonds towards 3' end. These nucleases are isolated from E. coli. Bal31. These nucleases cuts phosphodiester bonds towards both the ends, i.e. 5' end and 3' end. These nucleases are isolated from Ultramonas species. In this slide, we can see a picture which represents mechanism of nucleases. First picture represents lambda exo, where the nucleases cuts from 3' to 5'. Second picture represents exo3, where the nucleases cuts from 5' to 3'. Third picture represents Bal31, where the nucleases cuts from both the ends. Endonucleases. These are the other type of nucleases which cuts phosphodiester bond in the middle or towards the middle of DNA strand. Endonucleases are of two types. For single strand DNA, for double strand DNA. For single strand DNA, these are of two types. S1 nucleases, Mung-Bean nucleases. For double strand DNA, these are of two types. DNases, restriction endonucleases. For single strand DNA, S1 nucleases. S1 nucleases cuts phosphodiester bond from 3' to center. Mung-Bean nucleases cut phosphodiester bond from 5' to center. S1 nucleases are isolated from Aspergillus species. Mung-Bean nucleases are isolated from Mung-Bean plant. For double-strand DNA, DNases, restriction endonucleases. These are the enzymes used for double-strand DNA strands for cutting the DNA. In this slide, we can observe two pictures. First one represents the mechanism of S1 nucleases, where the two single-strand mRNAs are trimmed to same size from the 3' end to center. And the second picture represents the mechanism of Mung-Bean nucleases, where the two uneven strands of DNA are trimmed to same size from 5' to center. Polymerases. These enzymes synthesize or join new nucleotides to DNA strands. They are mostly used in replication process. Polymerases are of four types: DNA polymerases, RNA polymerases, T4 and T7 polymerases, Th polymerases. DNA polymerases are of three types: DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. DNA polymerase 1 and DNA polymerase 3 are ideal polymerase enzymes. DNA polymerase 1 has 5' and 3' cutting and synthesizing exonuclease activity. RNA polymerase is used for RNA synthesis. T4 and T7 polymerases are used for construction of viral DNA or RNA. Th polymerase has both RNA and reverse transcription polymerase. Th polymerase was isolated from Dermis dermophilus species. Polymerases are mainly used for synthesizing the nucleotides and phosphodiester bonds. Slide, we can see two pictures. The first picture represents the mechanism of DNA polymerase and the second picture represents the mechanism of RNA polymerase, where in the first picture DNA polymerase actions and synthesizes two double-stranded DNAs from one single-strand DNA. In the second picture, RNA polymerase synthesizes another RNA from one single RNA. Ligases. These are enzymes which join the gene of interest fragments from different sources. Mostly, ligases are used to join fragments of DNA by joining phosphodiester bonds. Ligases are of two types: DNA ligase, T4 DNA ligase. DNA ligase is used for cohesive end ligation. T4 DNA ligase is used for blunted ligation. T4-DNA ligase is extracted from T4 bacteriophage. DNA ligase is extracted from E. coli. This slide represents the mechanism of ligases. The first picture represents the mechanism of T4-DNA ligase, which ligates blunted fragments. The second picture represents the mechanism of DNA ligase, which ligates cohesive ends. The third picture represents the ends or type of ends present or formed during the digestion. Transferases. These enzymes add nucleotides towards 3' end of DNA strand. These are also called as terminal deoxytransferases. We can modify the cohesive ends to blunt ends using these transferases. We can add any nucleotide of our choice like ATGC, adenine thymine guanine cytosine, at the 3' end of DNA strand. In this slide, we can see the mechanism of transferases or terminal deoxytransferases. In the both pictures, the mechanism of transferases is mentioned that cytosines are synthesized at the 3' end of the DNA strands. DNases. These enzymes degrade or destroys DNA. DNases are of three types: DNase1, DNase2, MNases. DNases are used to degrade DNA, and these enzymes perform random degradation because they are not specific to recognition sites. DNase1 is little specific in cutting from 3' end. DNase2 is little specific in cutting from 5' end. But DNases are enzymes which cut randomly. MNases are isolated from Micrococcus family or species. DNases are mostly used to cut strands when huge DNA sample is present. In this slide, we can see the mechanism of DNases, where the first picture represents the double-stranded DNA is fragmented or destroyed into small pieces by using DNase1. The second picture represents the DNA which has DNA binding protein is fragmented or destroyed into small pieces by using DNase1. RNases. These enzymes degrade or destroys RNA. RNases are of two types: endoRNases and exoRNases. EndoRNases is of two types: RNase A and RNase H. RNase A is the enzyme which digests or destroys the single-strand RNA and specific towards two types of bases like cytosine and uracil residues. RNase H is the enzyme which digests RNA hybrids or DNA RNA hybrids will be unwinded by using RNase H. ExoRNases are of two types: RNase1 and RNase2. RNase1 cuts or degrades RNA from 5' end. RNase2 cuts or degrades RNA from 3' end. In this slide, we can see the mechanism of RNases. The first picture represents the mechanism of RNase H, where the DNA-RNA hybrids are unwinded and the RNA is degraded. In the second picture, the mechanism of RNase is mentioned, where the double-stranded DNAs are freed from single-stranded RNAs by digestion. Kinases and phosphatases. Kinases are the enzymes which add phosphate group at 5' end. Kinases are also called as polynucleotide kinases. Phosphatases are the enzymes which removes phosphate group at 5' end. Phosphatases are also called alkaline phosphatases. In this slide, we can see the mechanism of phosphatases and kinases. Kinases add phosphate group to the DNA or protein. Phosphatases removes phosphate group from the DNA or proteins. Restriction endonucleases. These are also called as molecular scissors or restrictases. These comes under the family of nucleases. These are discovered in bacteria in 1962. These enzymes are specific towards double-strand DNA. They cut double-strand DNA at or near to specific recognition sites called restriction sites. Restriction endonucleases are of three types. Type 1 restriction endonucleases. These are the enzymes which cut 1000 base pairs away from the recognition site. Type 2 restriction endonucleases are the enzymes which are also called as accurate enzymes which cuts at the restriction sites. Type 3 restriction endonucleases are the enzymes which cuts 24 to 26 base pairs away from the recognition sites. All the restriction endonucleases follows two rules. First one, for circular DNA, N rule. For linear DNA, N plus 1 rule. In this slide, we can see the mechanism of restriction endonucleases. The first picture represents the mechanism of EcoR1 and the second picture represents the mechanism of restriction endonucleases of HIND3, PST1 and EcoRV. This slide represents the difference between exonuclease action, endonuclease action and restriction endonuclease action. In this slide, we can see the total process of recombinant DNA preparation and screening. The first step is vector preparation, the second step insert preparation, the third step ligation, fourth step transformation and the fifth step colony screening. Thank you for your patience. Hope you have enjoyed this video.